Okay, so last time we were looking at the different characteristics of the microorganisms, including viruses. And remember, viruses are inert, but since you have to look at them under the microscope, they are considered as microorganisms, or studied within the world of the microorganisms. But then here we have the classification of life according to three domains. So we have the domain of bacteria that partially we already know about, and then archaea, and then eukarya. And eukarya includes protists, fungi, plants, and animals. So for this classification of the three domains, it includes several groups of microorganisms, and then they can be traced based on their ribosomal RNA into when they emerge, and this is the diverging of these microorganisms with time. So then within the bacteria group, we have some members like Thermotoga, gram-positive bacteria, proteobacteria, which are gram-negatives. You will see later on what is that. Cyanobacteria, and then we have within archaea hyperthermophiles, methanogens, and extreme halophiles, which is a functional classification. And then for this eukarya, we have some protozoans like Giardia, Glonozoa. We have as well green algae. We have some microorganisms that are called diatoms. We have ciliates, dinoflagellates. Animals are part of the eukarya world or domain, and then we have fungi, amoeba, which is a protozoan, and then we have fungi like slime molds, and then plants. Plant cells are also part of eukarya, and then what they're pointing here is that mitochondria, apparently, it was part of this domain, bacteria domain, and it got transferred into archaea, and then into the eukarya. And same for chloroplasts. And there is this reason to believe that because of the characteristics of the ribosomal RNA and having a specific ribosomes that are only exclusively found within bacteria, but the eukarya that has the mitochondria and the chloroplasts end up having these ribosomes. So we will see later on the classification of the ribosomes, and then we will explore more about this. Okay, now a little bit of history of microbiology. So in microbiology, we have several researchers that contributed to this field, including Robert Hooke, who in 1665 reported that the living things are composed of little boxes or cells. So he did observations on cork samples using a microscope, and then he saw square units. And these square units, he called them cells. Rudolf Virchow, he said that the cells arise from pre-existing cells, which means that basically we don't have pre-existing cells coming from decaying matter or dead matter, as many used to believe before. And then out of all these discoveries, early discoveries in microbiology, they came up with the cell theory. That means that all the cells, or all, living things comes from they are composed, of cells, and they come from pre-existing cells. And then we had the creation of some rudimentary microscopes, but they were really useful by a merchant of the Netherlands, whose name was Antoine van Leeuwenhoek. And he started to use these microscopes that he produced, and then started seeing microorganisms, and he did some drawings. He was actually a merchant that used to travel through Europe and then sell fabric. But in his spare time, he made some microscopes. And then these little microscopes, we have a replica now today, and this is how it looks. Now here, what you can see in this left side is a scale in centimeters. So it's starting from 0 up to 9 centimeters. So basically it's a little bit over 3 inches, the size of this replica. So it's something like this, more or less, the size of this little microscope. You have seen microscopes before in the lab. And can you imagine someone trying to look at tiny microorganisms using this microscope? It's incredible. So he polished some lenses, and then he put them in this brass plate. He made a hole in the middle, put it in the brass plate, and then he made all these little screws and the controls, like the focusing control, and then the stage positioning screw. He made like an opening here to make these to go up and down and front and back, so that with this little pin, which is the pin where he put the samples, he can see clearly through this lens the microorganisms. Things that he studied was water in ponds, and then even scrapings from his teeth. And he was successful at seeing these microorganisms through his microscope. He made the drawings, and he called them animalcules. Animalcules, basically like little animals. And actually his drawings are preserved up to now in some museums. Now, someone who also contributed to the field of microbiology was Edward Jenner. He was a physician that he was in charge, or well, he was responsible for producing immunity out of scrapings from the udder of cows. So he was working in a rural area, and he saw a lot of cases of smallpox. And smallpox it's a disease of the skin, but also can affect the nervous system, and it can kill persons. Up to the 1960s, early 70s, there were still some cases of smallpox in the world. This disease, thankfully, has been eradicated. And it was eradicated through a lot of efforts for vaccination. Now, smallpox is produced by a virus that is similar to a virus that affects cows. And this smallpox, when it infected the skin of persons, it created like little pustules, like raised lesions in the skin with filled liquid that hold the virus. And those pustules basically burst and then spread into the skin of the person, creating more lesions. And then it's similar to chicken pox. But basically, these lesions leave a scar, But smallpox, unless someone scratches pretty bad, you will have scars in the skin. But with smallpox, always leave scars. And the scars were so bad, and it was permanent, like a scar, okay? Now, people can live with scars, and we live with scars, even from a wound. It doesn't matter. The problem is that when that virus infected the nervous system, the person will die. So the mortality rate and the infectious rate of this virus is pretty high. And thankfully, again, we eradicated that. But in those days, when he was a physician, like around the 1700s or 1800s, basically there was no vaccination available. So he observed in milkmaids that whenever they were having a case of smallpox, the milkmaids, either they had it very mild or actually didn't get it. And the reason for that, it was because as the milkmaids were milking the cows, like touching the udders, if the cows had cowpox, which is a cousin, it's very similar to the smallpox virus, basically the skin of the hands of the milkmaids were pretty dry, and they had cracks in the skin, in the hands. So part of the virus from the udders, from the cows, went into their skin through this wound, and then went deeply into the immune system, and then they developed immunity. So they had a trained immune system then against cowpox virus, which protects them against smallpox. So he was clever enough to have this observation, and then he started to wonder what to do with the rest of the population. So then he grabbed the skin of the udder from one cow. I think it will have been kind of very brave to do that. And he scraped a little bit of the skin. He put it in a little bit of water, make like a homogeneous mixture, make a scraping in the skin of a child, put this liquid into it, and guess what? The child didn't acquire smallpox. And that's how vaccination started. So the term cow in Latin is vacca. So vaccination comes from this term. So then the protection acquired from vaccination, or even from having a disease, of course, if that disease allows you to, infectious disease allows you to improve your immune system, it's called immunity. Okay, well, not all the diseases are preventable through vaccination. What about those ones that it can't, and then that you have them? Well, you can have treatment of these diseases with chemicals. And any chemical that we take, any drug that we take to treat a sign and symptom or a disease is actually called chemotherapy. Doesn't necessarily need to be cancer. Like if you have headache, cephalea, and then you take a pill, like an aspirin or acetaminophen, any analgesic, that's chemotherapy. So, something that later on, like in modern microbiology, like in the 1900s, happened is the discovery of several antibiotics that are actually used to treat infectious disease. And these antibiotics can come from natural source. Mostly it's from bacteria like Streptomyces, or they can come from fungi. In the natural environment of these microorganisms, they produce their own antibiotics, but it's not for treating an infection. It's just to prevent the growth of other microorganisms around them so that they can have an advantage. This is called antibiosis, this property. These chemicals that they release kill other microbes or inhibit them from growing. And this property of antibiosis is actually the principle through which we produce antibiotics nowadays, either by extracting them from natural sources like fungi or bacteria, or by producing them semi-synthetically or synthetically. So those natural antibiotics, we can modify them. They will be called semi-synthetic, or you can plainly produce them from scratch without the aid of any microorganism. In this case will be synthetic. We will have a specific chapter dedicated for antibiotics, so you will learn a little bit more about this. Any questions? So then, an example of how these antibiotics were produced. So in the early 1900s, Alexander Fleming, who was a researcher doing microbiology experiments in Staphylococcus aureus, he grew Staphylococcus aureus in little containers that you will see later on in the lab that are called petri dishes. These petri dishes have in the bottom part like a layer of gel-like material that is nutrient for this bacteria. And then whenever he was inspecting his petri plates with this bacteria, once in a while he observed that some bacteria did not grow as usual, meaning that it was smaller than usual. And he started to wonder why. Well, every time that he saw these small groups of bacteria that we call colonies, they were always surrounded by a bigger colony. And that bigger colony were actually a mold that it was contaminating his petri dishes, and then these produce these antibiotics that basically kill or make this bacteria to grow less bigger than as it's supposed to be. So then he discovered penicillin, which was the product of fungus that is called Penicillium, the genus, and Penicillium, the species was Notatum. And in the 1940s, this penicillin was produced in mass production, and then it was purified, and it was used to treat bacterial infections, and it was successful. Now, there was another type of antibiotic earlier on that it was used, but it was not precisely antibiotic, it was a chemical that it was arsenic. It was called Valsartan. This arsenic was used to treat gonorrhea infections in soldiers, but it had like a toxic effect. So arsenic, it's a poison for us. So, but it was given in small doses. So then, when penicillin came to treat infections, it was kind of a good development. And here is the one example of antibiosis. So this is kind of almost replicating what happened with these experiments from Fleming. So this is the top view of a petri plate. So these petri plates are round. They are of different sizes, but mostly like this. This is the actual size. And then when you see it from the top view, you can see these little groups of bacteria. In this case, this is bacteria that is Staphylococcus. So this is the normal size. And then in this area, you don't see as many colonies of the normal size. You only see tiny ones, very, very small, And then you see this big colony, irregular shape. Well, this is penicillium, sending this penicillin, this chemical, to destroy the bacteria. So that's antibiosis. It's like an antagonism. Now, the description that I told you about Fleming and also about Jenner, Edward Jenner, basically tells you that observation is very important. I mean, how many times we might be apparently doing a routinary work, and then because of this routine, we miss the opportunity to see something that maybe was out of the routine. Well, I'm so thankful that Fleming was clever enough to see what is this instead of throwing away the plates and then start over the experiments. Why? Because thanks to him, we had the discovery of antibiotics. Now, part of the modern developments in microbiology includes the creation of branches of microbiology. Like we have bacteriology, that study bacteria. There are some researchers that only dedicate their life to study bacteria. We also have the study of fungi, which is called mycology. Study of viruses, virology, a study of parasites will be parasitology, and immunology is the study of the immune system. And immunology is highly linked to microbiology because we try to develop vaccination or new treatments for infectious disease, and then we study if the person develops a strong immunity or not, okay, with the aid of this. So an example of what immunology is doing, well, it is doing basically the creation of vaccines and molecules that we will see later on that are called interferons that are important for fighting viral infections. Now, we have a specific branch of science that is highly linked to microbiology and other fields of science. So this is recombinant DNA technology, which includes several branches like microbial genetics. Sometimes it's just not only, knowing the shape or the type of microorganism, but also knowing what genes it has that makes them perhaps so aggressive, or maybe why they are so mild. To study pathogenicity, the ability to produce disease. So microbial genetics includes studying how microbes inherit these traits, how they can transfer this gene to other microorganisms. And then molecular biology, study how DNA directs protein synthesis, among other things. Genomics, study the entire genome of an organism. Not necessarily has to be a microorganism. We have already studied the entire human genome a few years ago, and then it's important to know because knowing what the genes do, we can predict the consequences when these genes are mutated, if that makes sense. And then thanks to this branch of recombinant DNA technology, which by itself is called recombinant DNA, we had a very good development for the treatment of diabetes, which was the creation of insulin. So with this recombinant DNA technology, in the 1960s, Paul Berg was able to insert a human gene into a bacterial genome. So if this is bacteria, like let's say E. coli. E. coli, this is the chromosome. Sorry about the markers. So this genome, this chromosome, has places where you can cut them and open it up, which are called restriction sites. So you can cut, and then with that cut, you will have the ability to add genes. So in this case, he added the gene that help us to produce insulin. And he created a hybrid. So here we had, like, let's say the insulin gene inserted. And then this bacteria can be made to replicate, and it's going to make messenger RNA that is going to be able to be read so that you can produce insulin. And this insulin was purified and injected into people who couldn't make insulin on their own. So this is nice because before this, unfortunately, they had to use insulin from animal sources like pigs. They extracted the pancreas from pigs. They extracted the insulin from the pancreas. And although they tried to purify it as much as they could, the allergic reactions that produced were so bad. And not only that, I mean, the amount of insulin that you had from several pancreases was very little. So with this type of discovery, recombinant DNA, you can make a lot of that because bacteria can grow in high masses in little flasks with media. And they can produce a lot of the products that you want. And it's much easier to handle, of course, than animals. You don't have to sacrifice as many animals. So this is an example of this type of developments. Now, modern uses for microbiology in its field. So microbial ecology. This field is important because it helps us to clean our environment from either accidents or from normal activity that we have as humans. So for instance, we have some bacteria that can recycle carbon, nutrients, sulfur, and phosphorus that can be used as fertilizer by plants or can be added into the food of animals so that they can thrive. Also, bacteria can be used to degrade organic matter in sewage. So in water treatment plants, they have these bacteria that can degrade deposits that we have in the sewage that otherwise they will degrade maybe some of these products, but some of them, if they will degrade, it will degrade so slowly. And then some spills that has happened in oceans, oil spills or mercury spills, has been cleaned with time with these microorganisms. So these accidents that happens when you have an oil spill in the ocean, it affects many species. The fish that it's in the oceans, algae, all these little animals that live in there, including some of these birds that goes and feed into these fishes from the ocean. They can go and crash over these oil spills, and they end up with oil all over their feathers. So here what you can see, it's a water treatment plant and how it's separating the solids from the liquids and then use bacteria to degrade these contaminants. Another use of microbiology is the field is the creation of biological insecticides. So some microbes can affect insects, but do not affect the crops, and in this case can be added into these crops so that when the insects come, they ingest these chemical pesticides and then they die. And then you don't have the breeding of these animals, of these insects that can damage the crops. An example will be Bacillus thuringiensis. These produce diseases in insects, but is harmless to animals, including us, and then plants. Biotechnology, we use it to produce food, to work with the fabric that makes our garments. For instance, here we have an area where they are producing cheese, and they use microbes to give flavors to the cheese. It's really nice, right, that they are wearing PPE. They're wearing lab coat, hard hat in this case, and then in here, after the product is done, they're using hair nets. Do you see something there that you don't like? Because eventually you will develop that with me. What you don't like?
No.
No gloves. Who guarantees that these persons wash their hands whenever they were touching these items? I don't see. I'm opening the eyes for you. So I already kind of explained the process for this recombinant DNA. Recombinant DNA is not only basically using bacteria so that you can insert genes and then produce certain drugs, but also you can have fungi or even sometimes viruses useful to produce vaccines and enzymes. Okay? So many diseases that we have as humans are related to defective genes. These genes can be mutated or they can be defective. And with this recombinant DNA technology, they're trying to repair this. So they're trying to modify these genes so that, or add them into the genome so that we don't have diseases. So those were fields associated with microbiology. Any questions? And their uses? Okay, so as I told you last time, so we have more bacterial cells or microorganism cells in our body than actual human cells. And there is a purpose for this. It's not like we are living bioweapons. We have these microorganisms actually to protect us. These microorganisms, when they are found in several places in their body where they normally are found, like in the skin, the mucosa, like the oral cavity, the vagina, the intestinal lining, like the anus, like the colon, it is for a reason. Thankfully, most microorganisms are beneficial, but for those that are harmful, if they get in contact with us, they can produce disease. So we need to have a protective layer, and that protective layer comes from the normal microbiota. Sometimes it's called normal flora, and it has a variety of shapes and sizes of these microorganisms. So what you can see here is the lining of a tissue with microorganisms that can be long, like these rods. These ones are curved shaped. These ones that are sphere-like shape. And they will occupy receptors in our skin, in our mucosas, that otherwise the pathogens will occupy to produce a disease. Hopefully the dye from these markers went down. So areas of our body where we shouldn't have microorganisms. Will be blood, it's frustrating. You can help me with this. Where else we shouldn't have microorganisms? Lower respiratory tract. Body fluids, where? Want me to call you? What other body fluid we shouldn't have bacteria in it or microorganisms? Urine. Where else? What is the name of the fluid that bathes our brain? Cerebrospinal fluid. I know that you don't want to look at me because you don't want me to call you to tell me answers. I know that you're there, and you know that I'm here. Name of the fluid in between our joints. Shall we have bacteria in between our joints, the joints for the limbs? So synovial fluid. And then, of course, you can expand in places where we shouldn't have, like in our intestines, like the small intestines, etc. But basically the stomach. But the rest of the body essentially can be colonized by this normal microbiota that is beneficial and is protective. However, something to keep in mind is that these ones can enter deeper into our body through a wound, gain entry into the circulatory system, and then not become a normal microbiota. In this case, you will call it a pathogen, opportunistic pathogen. Even the normal microbiota can be in the wrong place, and then it can infect and produce problems. So functions of the normal microbiota: preventing the growth of pathogens. Some of them help us to produce chemicals like vitamin K. E. coli, it's an example. So it helps us to produce some groups of vitamin B. And then basically the property or the ability to fight off infections is known as resistance. So in our body we have several factors that help us to prevent disease, like physical barriers, like skin, mucosas, when they're not wounded. We have chemical barriers like the stomach acid. We have antimicrobials in our saliva, in our tears. The stomach acid is an antimicrobial. So that helps us to prevent infections. And we will see in the immune system classification of all of these factors. So I don't know if you know, but babies, as soon as they're born, they get a shot of vitamin K. Who remembers why vitamin K is needed for, or for what is needed in the blood? It prevents you from having what? It's important for the development of platelets, right? And platelets help us to prevent what? If you have normal levels of platelets, what will happen to your body? You need to talk to your professor. Prevent bleeding. So vitamin K is needed for having healthy platelets. Now, vitamin K, it is produced by bacteria in our intestines. Thus, a newborn has bacteria already in its intestines. Yes or no? When you were born, did you have a bunch of bacteria in your intestines? No. So there is this traumatic event that we all went through if you're born through birth canal. It's just passing your head through this narrow space, and that can create hemorrhages in your brain, ventricular hemorrhages. So then vitamin K is given to the babies so that as they start acquiring bacteria and as they start producing vitamin K, they already have vitamin K to protect them from a potential hemorrhage after they're born. Does that make sense? Yes, it makes. So then it takes around 8 days for the babies to develop the ability with these bacteria, because they start eating, and with this food that they ingest, bacteria will go and colonize their intestines, and then they will start producing vitamin K. Now, microorganisms, they form groups that we call biofilms. These biofilms form layers of these microorganisms that will be found in several places, including water pipes, including the bottom of the oceans, lakes, and our teeth, and tile in the shower, or bathtubs, walls, catheters. They can be found everywhere. And these groups protect each other. So basically it's layer over layer over layer. And then they try to find nutrients. So in this case, we have an example of a biofilm forming in this surface. It can be a catheter. Of course, when someone introduces a non-contaminated and sterile catheter into the human body, in this case, well, if the person followed an aseptic technique, they just clean around the area where it introduces this catheter. The catheter is not going to be contaminated. But eventually, with time, the skin of the person is going to be around this catheter, of course, in the base of the catheter, and it can go and colonize this catheter. That's why once in a while, these IV lines are replaced, or, of course, the one that is most likely to get contaminated is the one from the urinary tract. If it's not replaced, the person is going to have an infection of the urinary tract. It's not fun. By itself, having a catheter in the urinary system, it's painful, or, well, it's uncomfortable. But imagine having the urgency, the burning sensation of urinating through a catheter. And then, of course, if it goes bacteria into the bloodstream, that's not fun. Not at all. The person can die of an infection in the bloodstream. So that's why whenever someone is introducing these catheters into the human body, it has to be super careful. And even protect the area so that the catheter won't be as easily contaminated. So following aseptic techniques and steps. So then something that happens frequently is the overcoming of the resistance of the immune system by the microbes. And in this case, when this happens, you have a disease. So we have lately, more than before, more emerging infectious diseases, or EIDs. These EIDs are either new diseases or diseases that are old, but they are resurging, meaning like they are increasing in number in a short period of time. And this is related to several factors, including the immune system of the population. It is getting less stronger due to poor nutrition, due to exposure to a lot of stress, due to the lack of normal levels of sleep. So all of those can contribute. Plus, add that factor that we have less vaccination rates, and then maybe we are less careful when it comes to hygiene, to washing our hands, or whenever we're coughing or sneezing. There are some people that plainly, they don't even do this. Go look down, and then if of course, hopefully they have something to not put the microbes directly to the skin in their anticubital area. But the minimum thing that you should do if you don't have a Kleenex is just do this. Because many people, they just, whenever they cough or sneeze, they cough directly. Or they cough into their hand. I have seen that a lot, and my gosh, I get grossed out. They go and touch a lot of objects. Examples of EIDs: influenza, avian influenza A. So it's primarily found in waterfowl and poultry. And these birds are sometimes raised in farms, and many people don't use PPE, as we saw before, even when doing food, making food. So you're supposed to be very careful when handling these animals, because some of them, they can have the flu, the bird flu, and then this bird flu can infect humans or other animals like pigs, because we have all of these 3 species receptors for the same virus. And this virus, the genome, can acquire genes from other species. And then you have a reassortment of these genes, and then you have a new variant that can be more aggressive than others. So that's how the virus looks like. And then for EIDs, we have EIDs, emerging infectious diseases, in every single type of organism. So in this case, will be a virus. Now for bacteria, MRSA. MRSA, MRSA, stands for methicillin-resistant Staph aureus, which is a bacteria. Not all the Staph aureus bacteria species are MRSA. Only some of them. And the ones that are MRSA are resistant to several antibiotics, including methicillin. When they start to mass produce penicillin and they start to use it in the general population, in many people who were volunteers, the Staph aureus was very susceptible to the action of penicillin, was easily killed. But as time, the decades came by, some of these Staph aureus acquired genes that were resistant to antibiotics. They wasn't not killed anymore by penicillin. And they start to use methicillin, and then these became resistant to methicillin, these bacteria. And that's why they call it MRSA. And they start to use new antibiotics, much stronger, and then we had additional genes from the bacteria that makes it to be resistant to the action of different antibiotics. So here is kind of more or less the outline of the time when these bacteria became resistant to several antibiotics. Like in the 1990s, they started to use vancomycin, and then you had 2 strains that developed after this, like vancomycin intermediate Staph aureus or vancomycin resistant Staph aureus, or VRSA. And that's an example because we have other bacteria and fungi that are resistant to antibiotics. West Nile encephalitis. This is produced by virus, West Nile virus. And this West Nile virus was introduced into America by bringing birds in boats coming from Africa, from Uganda. So it was first, this one was actually, never mind. So this virus was discovered first in Uganda, in West Nile region, in 1937. And then some birds that were in contact with the insects that carry this virus were basically the carriers of the virus, because the virus took, the insects took a blood meal from the virus, from the birds, and then infected the birds. And then the birds came in a boat to America, and it appeared in New York City in 1999. And these were non-migratory birds. So then several birds were carried through maybe transportation within the continental US, and now this West Nile virus is found in 47 states. So modern transportation plays a role in bringing infectious diseases to areas where they weren't found before. Now we have an outlier in terms of a pathogen. So we know that proteins are part of the human body, like part of our bones, part of our skin, part of our hair, etc. But who would imagine that proteins will be infectious? So we have a specific type of proteins that are called prions. And prions are basically what it causes some diseases that are called spongiform encephalopathies. These prions can infect the human brain, the neurons, destroy the neurons, and make the human brain look like a sponge. Sponges have holes in it, so the human brain will have these holes in it. So then, one of these examples of emerging infectious diseases is Creutzfeldt-Jakob disease, or CJD. So this can be acquired through ingestion of cattle that has these prions, or also can be acquired by having a surgery with instruments that are contaminated with these prions. And another example of this is called the mad cow disease, because when it infects the brain of cows, the cows behave aggressively. It was discovered in the 1990s, this disease, and now we know what it causes, and then we know ways to prevent it, including not eating the meat or products of animals that were infected with prions. We will see more details about this later on, but at least in here, something that you can see, and this is a sample from a human brain that it is from a person who died. So this is holes in this brain tissue. This is not normal. And these are precipitated proteins within the human brain. So we will see later on how this is formed, but basically that's a postmortem sample from an autopsy. Another example of an emerging infectious disease is diarrhea, that is bloody diarrhea caused by a variant of E. coli, which is called E. coli 0157H7. Later on, we will see where does this nomenclature comes from. So this is an aggressive variant that can damage the lining of the intestines, and then the person will have bloody diarrhea. This It was first seen, this type of infection, in 1982, and it is produced by an E. coli strain that produces a toxin. So what you will see here, what you are able to see here, it's the lining of intestinal cells. So this is basically enterocytes, and this is a rod-shaped bacteria that is landing on top of this, and it destroys this enterocytes. This is the number one cause of bloody diarrhea or diarrhea in the world. E. coli O15787. It is acquired by getting in contact with food or water that is contaminated with this bacteria, and frequently is found in outbreaks, foodborne, in coming sometimes from picnics or from restaurants. Another example will be Ebola hemorrhagic fever that is produced by Ebola virus. Among the signs and symptoms includes high fever, hemorrhages, and blood clotting. Can it be contradictory? Yes and no, because there is this reaction of the body at the beginning in order to stop the hemorrhage that uses all these clothing factors, and then the person ran out of clothing factors, and then it will start having hemorrhage because it lacks these factors. Now, this virus infects the lining or the walls of the blood vessels, which are called endothelial cells, and then destroys them. So the person will have a hemorrhage. So it was first identified near the Ebola River in Congo, and it produces outbreaks once in a while. And thankfully, they have been developing vaccination nowadays. That it's perhaps still under later trials, but it seems like it's promising, this development. And this is how this virus looked like. Here is a blood cell, can be a lymphocyte or a neutrophil, and then the virus, it has this elongated shape. It looks almost like hyphae from fungi. And all of these images, with the exception of the one of the human brain, which is light microscopy, comes from scanning electron microscopes that are very fancy, and they can enhance these images with color. And this is kind of a three-dimensional view. So these are really nice microscopes. Protozoans can also produce emerging infectious diseases. So we have a protozoan that is called Cryptosporidium, and there is a specific species, Cryptosporidium parvum, that can produce severe diarrhea, massive diarrhea. It's not bloody diarrhea, but the person loses a lot of fluids. And that can lead to acute dehydration and death in some populations. So it was first reported in 1976, and mainly is transmitted by ingesting contaminated water. So here in the United States. So it causes 30% of diarrheal illnesses in developing countries. If someone's immune system is strong, this can be self-limiting infection. But if someone has immunocompromised system, the person will not have self-limited illness. So a person can die of this. Actually affects people with AIDS. And something interesting about this protozoan is that this protozoan actually is resistant to chlorination. So the water can be chlorinated, but if someone contaminates this water, the next person that drinks this water or get in contact with this water will get cryptosporidiosis. Now, Here you can see the lining of the intestine, and this protozoan, it has cysts that looks like a sphere, and then it's a protozoan that it's actually also spherical. Now, this is small, and at least in certain pools, swimming pools, they use filters to filter the water, because most swimming pools, of course, they use chlorine to sanitize the water, but that's not enough. You need to filter the water to prevent these protozoans to be freed up in the water, to be free in the water and then contaminate to spread to whoever is in the pool. So I guess most of us has been in pools, and there is no one in a pool who never has drank a little bit of water. Okay? So imagine you being in a pool with a bunch of people, and unfortunately not everyone is very hygienic in the pools, so they can have an accident, and then those microorganisms can go into the water. And then you're swimming carelessly, thinking that nothing is going to happen. But then if that pool doesn't filter the water, only chlorinates it, well, if the water is contaminated with cryptosporidium, you can get cryptosporidiosis. So, you will hardly see me in a pool. Maybe never. Another emerging infectious disease. It might have some data that it's outdated, but it's acquired immunodeficiency syndrome, or AIDS. It's caused by infection with human immunodeficiency virus, or HIV. It was first identified in 1981, so we will see in the virus chapter a little bit more details of this. But worldwide, it's infecting around 33 or more people, and it produces around 7,500 new infections every day. It can be transmitted through several ways, including sexual intercourse, receiving blood transfusion with this virus, or it can transmit from mother to fetus, and also to breastfeeding. In United States, HIV/AIDS affects 26% females, and 49% are African American. So that's all for this chapter. Do you have any questions?